Well, we have a lot to cover today, and it's a couple minutes after 10. So if our presenters are ready, I'll get this thing kicked off. Trivia, are you with us? There you are.
Yes. Hi, Kristen.
Good morning. I'd like to start by welcoming everyone and thanking you for joining us today for this presentation, Human-Derived Hydrogel in a PDX Model. I'm Christine King with Certis Oncology Solutions, and I'll be moderating today's presentation. Before we begin, just a few housekeeping items. During the presentation, all attendees will be muted. We have a lot of time at the end of today's presentation for questions and answers, and you may submit your questions using that option at the bottom of your Zoom screen. If we're unable to get to all the questions during the webcast, we'll follow up with you after the event. The presentation will be recorded, and we will send a link after the presentation ends. Now I'd like to introduce our presenters, Dr. Trivia Frazier, the co-founder, president, and CEO of Obatala Sciences, who will introduce her company and discuss applications of hydrogels in general and human-derived hydrogel specifically. Also with us is Dr. Jonathan Nakashima, Chief Scientific Officer at Certis Oncology, who will present findings from a study his team performed comparing various hydrogels in a PDX model of lung adenocarcinoma. With that, Trivia, I will hand it over to you. Please take it away.
Thank you very much, Christine. It is a pleasure to to jointly present this technology and applications of our hydrogels, and also highlight some of the work that Certis is doing in the PDX area. And so we'll start off essentially, as Christine mentioned, by introducing Obatala Sciences, what it is that we do, our products and services, very quick high-level overview to give some context to the hydrogels and the specific hydrogel that we're presenting today called ObaGel. And then we'll look into dive into Certis Oncology Intelligence, essentially allow Jonathan to present their PDX model development and the comparison study using ObaGel. And so essentially what we do at Obatala is we manufacture human-derived hydrogels, the stromal stem cells, and stem cell supporting products. And what that means is essentially stem cells that are used to mimic within the context of three-dimensional cell culture and traditional two-dimensional culture, but in various applications, including mimicking tissue. And for us, that tissue mimic is strongly with the intent of taking it back to the human. So we're looking at how can we better mimic diverse patient populations for the identification of better compounds, better therapies to treat patients who need these therapies. We are addressing a priority that is established at the federal level to essentially use more human-like tools in the preclinical research stage. And what we're doing in terms of our bottom line and our goal overall is to reduce the animal trial size and cost by increasing the efficiency, increasing the amount of data that we get that's clinically relevant, and bringing it to the preclinical stage of development. And so at a high level, we have our stem cells that I mentioned, very highly characterized, high-performing cells that we sell at early passage from adipose bone and bone marrow. And the companion products include ObaGel, the hydrogel that I'll highlight in just a moment, that's used in the context of three-dimensional culture, but also in PDX model development, as we'll get to in the topic of today's webinar, and also the accompanying media products, which help to support the growth of stem cells, but also their differentiation along the adipogenic, osteogenic, and chondrogenic lineages. And then we provide all of those in the form of a kit. And we also provide a service to researchers who are interested in establishing the ObaCell, which is the tissue model in which culture cells in 3 dimensions and looks at functional outputs in response to different therapies. And so our hydrogels, I'll highlight and hone in, move to ObaGel as one of them. They're tested in the context of their ability to support multiple cell types. And so at the initial baseline that we look for is biocompatibility with stromal stem cells, which is within our core expertise. We are sourcing the material from human tissue. We're looking at consistencies from batch to batch. So we want to and provide that assurance of performance from lot to lot. And we're looking at providing products that have tunable biomechanical properties so that you have a range of stiffness and viscoelastic properties that are suitable to the specific tissue that you have in mind. And our product ObaGel is specifically a thermoresponsive gel that retains its liquid status at 4 degrees and gels at 37 degrees. And so the optimal tissue construct, or in this case, tumor supporting construct, is one by which we don't lose volume. So the volume retention is persistent after long-term implantation and in vitro culturing. And so ObaGel has multiple applications. I sort of alluded to it a little bit earlier when I mentioned the application within 3D cell culture. And so it has been validated for that specific market in the way of mimicking human tissue that applies to adipose, bone, as well as vascularized versions of those. And we're looking at other tissue types to support, and have been approached in the context of liver, for example, fatty liver. And so we're also beta testing ObaGel in the context of CAR T development. So the expansion of CD34 positive cells that can then be used in the context of CAR T and CAR NK cell expansion. And then an emerging market for it, as it is a human blood-derived material, is within the context of pressure injury therapy, so clinical applications. In terms of characterizing this gel, as a start, we work with AABB accredited blood banks that we receive discarded medical waste. And that provides us with a starting material that is clinically suitable. And so our goal is to continue the manufacturing in a cGMP method that allows the clinical and therapeutic applications. Some of the properties that we have also described that I'll dive into in just a moment are centered around the protein profiles, looking at essentially what provides this enhanced cell growth. Again, reducing that lot to lot variability, but we're doing that by pooling. As you know, there are donor to donor differences, and we reduce those differences and maintain lot to lot consistency by pooling a minimum number of donors. We're looking at the growth factors and hormones in terms of maintaining a variable range by which we can look at the ability to mimic the in vivo environment more closely. And so the applications extend beyond the 3 that I mentioned into areas that we're interested in pursuing partnership in, and that includes bioprinting and bioink development, disease modeling, organoids, organ-on-chips, and what we're presenting today. And so we have published a comparison of ObaGel to Matrigel. As we know, Matrigel has been considered to be the gold standard with respect to tumor culture. When we look at human-derived cells and maintaining them, we again are looking to take it back to the human and away from the mouse and away from mouse tumor. And so Matrigel being derived from the Engelbreth-Holm-Swarm tumor, it is that standard example. And so we published this work in biomolecules using tandem mass spectrometry, and we followed that with eye tracking and quantification and performed the protein analysis, Panther. And what we see essentially that Matrigel is enriched twofold compared to ObaGel in cytoskeletal row GTPase proteins, transport and translational proteins, and inflammatory proteins, whereas ObaGel is enriched twofold or greater within a cell adhesion molecule, as well as protein binding and prothrombin pathway activating proteins, which is somewhat intuitive as it is a blood-derived material, but it is also upregulated in Wnt signaling proteins. And so what we have also been able to demonstrate both within our laboratories, but also through collaborators and entities that are now using ObaGel for their respective applications, is that it supports the stromal vascular fraction cells, endothelial progenitors, and other more mature and less mature endothelial cells, HUVECs, iPSCs for the context of generating an induced mesenchymal pluripotent cell, MPC. So in the context of bone osteogenic development, for example, and in the cancer area within the context of prostate cancer, breast cancer, endometrial cancer, even endometrial cells that are primary cells derived from patients, and then in today's application, lung adenocarcinoma. So not only is our hydrogel supporting multiple cell types and also a combination of what would be heterogeneous cell types within one system, but it is also being used in the context of both static and more dynamic environments. So in our laboratory, we use it within the context of 96-well formats or even other multi-well formats. And within the hands of our collaborators and other laboratories, it's used in perfusion bioreactors in the context of multi-layered perfusion chips and vascularized chips. And so when we establish those three-dimensional constructs, those multi-well chips, we are combining our primary human patient-derived stromal vascular fraction cells, which contain a combination of the adult stromal stem cells, but also some hematopoietic stem cells, some of the B and T cells that are local to and tissue resident within the adipose depot of interest, but also a number of other cells that help to provide the functional elements of adipose tissue and contribute to its own ECM development. We combine those stromal vascular cells with ObaGel and do a full encapsulation and allow those cells to then remodel ObaGel. And they form these very nice networks when they're placed within 37 degrees and allow to interact with the gel. And what we've demonstrated and also published in Stem Cells International is the ability for the gel to maintain a heterogeneous population of cells, but also to promote that endothelial subpopulation to support robust network formation. And that is demonstrated by our increase of CD31 expression, a cell surface marker for endothelial progenitors, along with CD34 in terms of a co-expression with freshly isolated stromal vascular fraction, and comparing that to 1 day after culturing within the gel and out to 1 week. We've even looked at this over a 28-day culture, and we can see a maintenance of the endothelial progenitor subpopulation. And so what we get is a hydrogel that has the ability to support multiple aspects of functionality within an ideal tissue model, including vascular network formation, which I mentioned with the endothelial progenitors, immune cell response, which can be provided by the B and T cells, but also in a more functional level, macrophages, which would be much more prevalent within an obese model, and the adipose tissue response in terms of functionality, glucose uptake and lipolysis. And what we've been able to demonstrate is that the hydrogel supports these efforts, and we can look at differences from patient to patient because the stem stromal population retains some of the key features that maintain those differences from patient to patient. And so the cellular combination itself is contributing to the stromal compartment functionality, which is supported by a robust system, and the output is a representation of diverse populations. What we have demonstrated is a functional spheroid type of culture. Also, what we're looking at is supporting organoid culture. In this context, this is an adipose differentiated spheroid. And as we increase the percentage or the concentration of ObaGel within the culture media, we get these denser and larger spheroids with respect to their diameter. And so that can be modulated essentially and adapted to multiple substrates in multiple formats. We also have been able to visualize our constructs, but also the network formation from an endothelial progenitor standpoint, the vascular response after seeding the cells within ObaGel within 2 weeks. However, we also start to see this network formation occur and start as early as 3 to 5 days post-seeding within ObaGel. And at the very bottom, you see scanning electron microscopy images. These are cryo-SEM images of the hydrogel alone in the left panel without any cells seeded. We see a difference in the remodeling and hence the porous makeup of the gel in the presence of cells, and then also in the presence of differentiated cells. And also in this context, leading up to the relevance of this particular webinar, we're discussing an in vivo application of ObaGel. One of those is for recapitulating an adipose depot in mice in vivo. And so we conducted a 6-week study by which we implanted ObaGel alone and ObaGel in the presence of stromal vascular fraction cells, human stromal vascular fraction cells. And we seeded those, implanted those in the subcutaneous dorsal sides within C56 Black 6 mice. And after 6 weeks, what we saw is the ObaGel control supported the migration and adipogenic differentiation of the host's own cells. In the presence of the stromovascular cells, so ObaGel seeded with the human SVF cells, we get a very nice depot that represents a human, a subcutaneous adipose depot with much larger nuclei as well as lipid, intracytoplasmic lipid droplet formation. And extending that, so after demonstrating, this is a very strong application for our hydrogel to support in vivo humanized depots within mice, we then looked at the possibility of supporting cancer PDX models. And in this particular instance, we collaborated with a number of researchers and were able to identify breast cancer as one of the potential initial applications. And so we published this as well in Tissue Engineering Part A. And we are demonstrating the different mechanisms by which a blood-derived material such as ObaGel can be used in this context. But for our particular application, we're looking at the decellularization of a tumor via collagenase digestion to yield either ASCs and the breast cancer cells individually, those cells that can be used in combination with the immune cells that are going to secrete the impacting growth factors and cytokines that influence the microphysiological environment that we're trying to recapitulate. Or in the case of using decellularized adipose tissue alone and reseeding with cells of interest, whether it is in the context of immortalized cell lines or in the context of primary cells, the end goal is to generate a more physiologically relevant depot that can be used and propagated either in vitro or within an animal model. And Certis has done a great job of developing their own PDX model. And so I'll turn it over to Jonathan now so that he can describe their model and the application with ObaGel.
Thank you, Trivia. Happy to be here, and thank you for giving us the opportunity to co-present with you. So what the audience is seeing here right now is one of our models that we've developed here at Certis. This is a lung adenocarcinoma model. This was your typical patient-derived xenograft. And what we did from here was we derived a culture off of that xenograft. You can see the information on the tumor on the left side here. It's a lung adenocarcinoma, come from a metastatic site on the patient's hip. And you can see the patient details on the bottom there and how heavily pretreated this patient was. We derived a culture from this model as well, and then transduced it with luciferase, so therefore we could study metastasis. So in this example here, not with ObaGel, this is a separate project, but we took these cells, we injected them into the hearts of animals and just tracked with BLI where the cells went. And as you can see, they populated the lungs, the kidneys, surprisingly the ovaries, and pretty much went pretty systemic in the animal. On the right-hand side is the histology verifying that the tumors were present within those organs that we saw and that they did stain with anti-human nucleoli, showing that they are human tumors of origin. So we took this model and then we did a study, sorry, next slide. So we took this model and then we used this and compared how engraftment in the subcutaneous setting would differ among ObaGel as well as kind of the 3 standard ECM gels that are available on the market. On the left-hand side is our methods. These were done in female nog mice. Again, it was a subcutaneous injection and we injected 3 million cells per site. We did 5 animals dual sub-Q for an N of 10. And then what we injected is depicted below. It's a 100 microliter injection. In your typical Matrigel ECM mixtures, we did a 1-to-1 ratio of the gel with the PBS with the cells, resuspended them. But with the ObaGel, we followed the instructions. We did a 3-to-1 mixture with the ObaGel and Obavate, and then we resuspended the cells in that mixture and injected it directly without mixing with PBS. In the middle graph is the mean tumor volume over time. You can see that all 4 groups have a pretty consistent growth rate within themselves. There was no statistically significant differences among any of the gels, and for the most part the graphs overlap pretty well. Obviously there's minor differences due to the variability, but you can see that on the individual graphs on the right hand side that the actual tumor growth of each animal. So by and large, we were pretty satisfied here that one, there does not seem to be a significant difference between any of the commercially available matrices, and 2, that ObaGel, being one of the new ones, was able to perform substantially well compared to the kind of standards in this practice here. So, you know, this is a pilot study. It's the first one. I think it warrants some more investigation in vivo. As Trivia has already mentioned, some other studies have been done. And I think that this is a great start, and it'd be interesting to see the downstream analysis on this. So what we're currently doing right now is histology to look at the differences, if there is any morphological differences within these tumors. And we've also taken samples of these tumors so that we can perform gene expression analysis to see if there's any significant differences in gene expression of the same model within using these different gels as well.
Thank you, Jonathan.
Thank you, Trivia.
And I'll stop sharing. Before we kick off Q&A, Christine, I apologize. I did not allow time for the poll. We did have a quick question so that we could get to know the members of the audience, and you know, mainly, do you use hydrogels at all within your research? And you know, if so, which ones have you used?
Just give a minute.
Yeah.
Lots of participation. That's always awesome.
Yes, that is awesome.
All right, it looks like we may have the answers we're going to get, so I'll end the poll. And then Trivia, do you see the Q&A there? That we did receive a question from one of the participants?
Yes, yes, I do. Thank you very much. Hi, Marxa. Thank you for your question. Marxa's question is, has anyone tried growing ASC in ObaGel, and do they survive or differentiate fat into fat? And yes, absolutely. We have tried not only within our laboratories, but also within some of our customers' laboratories and collaborators. They have used adipose-derived stromal stem cells, so ASCs, in ObaGel, both in a context of proliferation, so short-term as well as longer-term, 2-week, and even out to a month proliferation. And then induction along the adipogenic pathway. Endpoints that were investigated within those studies were functional endpoints, so glucose uptake and lipolysis, but also adipokine secretion and other endpoints of functionality from a adipogenic standpoint. And they do not only survive, but grow very robustly within ObaGel. We'd be happy to share that publication with you as well. I didn't dive into that data for time purposes today, but it is within Stem Cells International. The lead author is Robert Bender, and it describes the growth, the impact on a proliferation, and the adipogenic differentiation lineage. Thank you for that question. We have another question here from Lawrence Bernetti. How fast does the ObaGel deteriorate in your mouse studies? Well, that's an excellent question. And so I could talk about our experiences within our laboratory, and then I don't know, Jonathan, if you did observe any of that within this context, but I'll kick it off. Essentially, ObaGel, in terms of deterioration, what we didn't measure difference with respect to resorbents in terms of that equivalence. So at the initial time of seeding versus implantation rather, and 6 weeks post, we did measure the sizes of the depots that we generated, but did not say this is a margin that corresponds directly to the hydrogel. And so, in the absence of taking that direct measurement of the hydrogel beginning in post with a labeled, preferentially a labeled version of the hydrogel or that of the cells, we're unable to speak to deterioration within the mouse studies, but that's an excellent question. One that we are interested in pursuing and learning further. So if you are interested in trying the hydrogel for a pilot study, we would welcome that collaboration.
I'll just add to that from Certis' side, we did not look into this. If you recall on the graph, we let the animals, the study ran for a period of about 90 days or so. So we just implanted, and then we just were mostly concerned with tumor volume over time and see how the kinetics changed between the different gels. So there were no time points where we took down animals and analyzed the amount of gel that was still present.
Okay. Thanks, Jonathan. And it looks like we have the results as well from, let's see, from the poll. And 88% of our viewers use Matrigel, followed by a tie, a three-way tie. Well, a two-way tie between Cultrex and Geltrex. So thank you very much. That's helpful information with respect to what is currently being used for your existing studies. And this was 8 people. Thank you.
We should have had another poll, and that was to ask how many people were impacted by chain supply of Matrigel in the past few months, because Certis has experienced that, and I can say that a lot of other institutions has as well. So this was one other reason why we wanted to do the study, to see if there's a difference and if we can shift and give us the confidence if we need to pivot given resource constraints with global supply chain.
Yes, excellent point, Jonathan. Thank you for bringing that up. That has also been the context of many conversations that we've had recently, supply chain issues with Matrigel and other gels that are being used for similar applications.
Trivia, can you speak to about your how available ObaGel is as far as supply chain goes and whatnot?
Yes, absolutely. So we have ample stock of ObaGel ready, packed to ship, to provide to customers. And that's partly because of the source material. Just like our cells, we use what's routinely discarded medical waste from human. So it would either be blood collection centers or in the context of aesthetic surgical procedures or orthopedic procedures like total knee replacement, total hip replacement, those types of procedures. And so because of that, the unfortunate thing is also fortunate for the research side. It's unfortunate that we continuously need to have these types of procedures done from a bone and tissue replacement standpoint, but that also provides us with ample supply as a starting material to manufacture our products. And so we're ready to ship out in the context of interest of ObaGel.
All right, fantastic. Well, it looks like we've answered all the questions that came through during the webcast, covered a lot of ground. Thank you both so very much for your time this morning. Thanks to everybody for joining. Just a quick reminder, you will be receiving a follow-up email and a brief survey, and we do hope you'll take a few minutes to share your feedback. Thanks everyone, and have a great rest of your week.
All right, thank you.